All right, so hello, everybody. Welcome to the North American 3Rs collaborative webinar series from the Translational Digital Biomarkers Initiative. And as Matt already introduced, today we're going to talk about optimizing study design. Next slide, Matt, please. So if you're not familiar with North American 3Rs Collaborative, which I'll be surprised if any of you guys are not, but here's a website that you can go to check us out. Also, if you have any questions that you'd like us ask us, you can contact us at North American 3Rs. And then so this initiative, this collaborative has an amazing board and team. Here's just some pictures from the board and other members. And as you can see, also the partners that are part of this collaborative. So, from regulators, from government, end users, technology providers, other nonprofit. We work very closely with NC3Rs. So again, it's an amazing group of people working on 3Rs and leading 3Rs initiatives. Next slide, please. So when we think about initiatives, we want to make sure that those initiatives bring strong evidence, so it allows folks to build big impact from those initiatives. And the third part, it's really imperative, is for us to being able to implement those initiatives and what comes out of those into day-to-day processes. So this is where we think about real-world practicality and making sure that what comes out of those initiatives, we're able to do that with various stakeholders. Next slide, please. So currently we have 6 really amazing initiatives, and they have been happening for various periods of time. We have the Rodent Health Monitoring Initiative, and you can see each one of these on our websites. Microphysiological Systems Initiative, Translational Digital Biomarkers, as you are aware of, since this is what we're presenting today, Refinement, 3R certification course, and also compassion fatigue. Next slide, please. So for each initiative, we want to bring in thought leadership, so where we can come up with a consensus from various stakeholders, bring real world experiences, and research the science to back that up. We also look at benchmarking and how people feel about the different initiatives and the impacts that's driving, looking at barriers for that, for what comes out of these initiatives and how we can implement those. And one of the biggest aspects is also education and training and providing practical resources that you can take away and bring them into your own organizations and share that as you learn about the different initiatives and what comes out of those. We also have an FAQs section as well that you can really utilize for each one of those initiatives. Next slide, please. So again, today, we're going to be presenting a webinar from the Translational Digital Biomarkers group. And the goal behind this initiative, so it's been around now for a few years, is really to increase adaptation and figure out regulatory acceptance of digital biomarkers. So also understand what are some of the challenges, what are their opportunities. So for folks that are working with those technologies, I think you're going to benefit from this webinar. But even if you're not, if you're just thinking about those technologies or learning about those, these webinars present an opportunity to learn and then potentially engage. One of the aspects that I do want to mention that if you are interested in joining North American 3Rs and each one of those initiatives, but specifically this one, please reach out to us and we'll be more than happy to have you engage. We're really looking to expand each one of those groups. Next slide, please. So currently, the Translational Digital Biomarkers Initiative has 17 members, and it varies from technology providers, end users, and regulators as well. And here are some of the organizations that are participating in this initiatives. And it's growing pretty much from every quarter to quarter. We also really want to collaborate and bring various stakeholders. We recognize none of these technologies and these initiatives will move forward without collaboration. That's what allows us to expedite these. And there we also identify the value and share how to really facilitate and optimize implementation of this technology. And then obviously the biggest goal is to promote the 3 Rs, as we can reduce the reliance on animals as well with these various initiatives. Next slide, please. So there are various resources that we have available. There's various manuscripts for the initiatives that we already have published. During the webinar, I'll actually put in a chat here and share with you the manuscript that we published last year. We also present at various conferences. We have webinars. We provide educational resources. And we have also developed tech hubs for the MPS initiative. And we're going to follow up with a tech hub for these technologies as well, which we'll share with you as when it goes live. Next slide, please. So again, today we have a fantastic group of presenters. So first, I want to thank them to taking the time and jumping in here. So we have Sheba Churchill from Charles River, Ellie Carlson from Calico, Angela King-Herbert and Jennifer Fostel from National Institute of Environmental Health Sciences. So with that, and also I just want to mention for questions, please put them in the chat, and then we'll address all of the questions at the end of the webinar, but we'll collect them throughout it. And also, I would like to thank folks that already submitted questions before the webinar. That was definitely helpful, and we'll address them during the Q&A at the end of the session. So with that, again, welcome everybody, and I'll pass it on to Sheba and Angela.
Thank you. Just a second.
And I guess Matt,
you guys see my screen okay? Great. So, thank you guys for being here. Again, my name is Sheba Churchill, and I'm here presenting this particular portion with Angela King-Herbert. I'm coming to you guys from the user end standpoint. What we wanted to do was kind of introduce you to kind of 2 studies we're working on that we appreciate the help this group has given us in terms of. Can anyone hear me? Okay, good. Yes. Sorry. And just make sure we share some of the things that we've taken away from designing our studies, give you some of the challenges and opportunities we found within it. This group has been immensely helpful. Additionally, using the paper that has come out of this group has been just wonderful in terms of helping us kind of work through different challenges that we've had in trying to put our studies together. So I'll go through just quickly the 2 studies. The first study we're working on is called Evaluating the Use of Physiological Monitoring in the Inhalation Study with our Sprig Dollies testing to EP. So what we're doing here is we're looking at trying to inform, basically using translational digital biomarkers within studies to kind of look and see, okay, can we expose animals to a certain toxicant and see if there's any change in terms of capturing baseline core clinical measurements. Is there any impact to the housing conditions and how we're monitoring stress? And so this has a really good also additional animal welfare component of what we want to kind of see. Okay, are we seeing things that are changing as we're using different intoxicants in our studies? We end up choosing one of our more complicated studies with inhalation to kind of test this out in. And so additionally, another goal of ours was to look at the complexity in the data that was going to be generated from this and help work through this. And the study, we're working on it throughout the year, and we're this close to getting it into our vivarium, working through it. So another study we're working on is machine vision enabled behavioral tracking through cross species. This study, what we're doing here is we're trying to identify chemicals that are most likely to be neurodevelopmental, cause neurodevelopmental disorders. And what we're doing is placing it in a set of fruit flies, using AI to go through and comb through all this data that we're going to be using to extract from these studies. And then we will move the same chemicals over into a mouse model and then compare the two. And so this is going to be new for us. We're actually literally building the actual devices right now in-house and working through how to interpret all this footage and do a certain set of behaviors that we'll be monitoring and tracking to kind of better characterize these environmental chemicals. So things that we've learned going through this, when you're approaching this and you're looking at what kind of technology and things you may want to use, the key is making sure you have everybody in your group. And some people will be like, oh, it may be too many cooks in the kitchen, but it's so important to have every member of the team there at the beginning when you're trying to pick out this different technology, whether you're building it in-house or you're going to a provider to bring it in. Some of the basic things that we have looked through, we worked through with IT, we wanted to know, okay, do we actually have the ability to host this tech within our institute? Do we have the infrastructure? We needed a person on our team. Jennifer is wonderfully our person kind of for that and helping us kind of understand better what we can do. Also with cybersecurity, one incident we went through, we had this tech, we loved it, and we were like, oh, we're going to bring this in, this is going to work great, but found out the way their flow of data was, it had to go into their private cloud, and that's not allowed. And I would not have known that if we didn't have people on our team that were there to kind of pull in and say, hey, no, you can't do this. You can't really have too many cooks in the kitchen. You need to pull in Vivarium leadership. There are basic things like connectivity. Are there other people who are using this tech that I can piggyback off of things and changes they've made within the Vivarium? Will other scientific staff be invested in this? Will someone else want to use this? This is all important things you need to kind of know ahead of time when you're planning. And also bringing in your data scientists. How will the data be collected? How will it be stored, formatted? All those different steps that I know nothing about, and I would need someone there to help. Additionally, with the biostats person, we needed to know kind of what information do they need to help us with our statistical analysis at the end. So next, we kind of went into talking outside of the team. When you're working in a group as the lead, you always have to have an ID person that's your spokesperson, but they will help you funnel the people they will help you with your group as a leader to ask questions for your tech. They are the people who are going to schedule the group calls. They'll bring in vendors and early in the process to kind of help you weed through what you need for your particular study. Also, we went through and combed and looked for publications with similar tech. And also in talking to some of the providers that we were looking at, we ended up having a connection between a collaborator that was actually less than a mile away from our institute. And that was so useful. We were able to talk to that person about the pitfalls and successes they had because they had done multiple studies similar to ours, and we were also able to have them come in, do a seminar, talk through things. They brought in, we just basically kind of unfortunately held this person hostage for several hours, just asking questions. Okay, well, we have this. Is there gonna be noise in the data? Is this just little questions that you needed to know as we were kind of preparing to go into, if we're preparing to go into the study. And so additionally, you want to be able to, whether you're writing a grant, whether you're whatever portion that you're doing, you want to be able to identify questions and hypotheses or bring in key points where your TDB can add unique insight. You want to make sure to express the added values that you're bringing in when you're talking to stakeholders and people in your institution. Some of the things we had were better. We would have better insight into disease models, better insight into improving animal welfare, implementing the 3 R's concept, then decrease inter-experimental variability. Also, additional data that will be better to inform our traditional models. That's one thing specifically with our first project, we're looking to hope, okay, we've done this traditionally. What will this data, this new data that we collect, how will help inform what we traditionally have been doing? And so that's one thing we're really excited about and hoping for. So I'll hand it over to Angela.
Okay. Thank you, Sheba. Have the
Have
next
the
slide.
next slide. Okay.
Got it. So you want to consider a pilot or feasibility study. Um, no question, as Sheba said before, no question is too small or unimportant. The point is to ask as many questions as possible. And when you have a lot of folks on your team, that helps to think of the little things you may not have thought of before. Um, to You want to manage the risk while identifying deficiencies that can come up. And your resources are time, people, money. But in the long run, it's more efficient to have a feasibility or a pilot study because you are better prepared for the I'll say real study. It gives you a better idea of what's going to happen in the real study. Um, Preliminary data can give insight on the things that may be needed or things that may be missing when you are doing a pilot or feasibility study. Can I have the next slide, please? While you're doing this, there are a lot of questions that you should be thinking about as part of the study. Um, there will be important nuanced things that are not thought of. Procedures, husbandry care. What time will the data be collected? Will it be collected while there are technicians in the room? Should there be baseline data collection? What is the length of that baseline data collection? What is the recovery data collection? So lots of questions you need to kind of bring forward and think about as you're going through doing the studies. And remember to ask for outside help, collaborators or others who have done the studies before. They can really add a lot of information, insight that if you're new to a study using digital biomarkers, you may not quite understand all the nuances that you need to prepare for. Next slide, please. When analyzing the data, review the data in real time. I can't say that enough. Review it in real time or as close to real time as you can. So suppose you have a 28-day study going on and you have 60 animals and you're collecting all this information on these animals using the digital biomarkers. If you wait to day 28, you're going to be totally overwhelmed. It will be very difficult to sort out what you're really looking at, and it's going to take a lot of time. So I would strongly suggest that you look at the data um, within the next 24 hours. So the data is generated on a Monday. You want to start looking at that data on Tuesday, or have some sort of idea of when you want to review that data. I say real time. You're not really doing it in real time, but you're doing it as close to real time as possible. And you want to understand what the data output is going to be. So if you're looking at, for example, you're looking at uh, heart rate, or is the output going to be numbers when you're looking at heart rate, or is it going to be waveforms? Just know what the data output is going to be from the start of the study to the finish, just so you could better understand what data you're getting. And our Dr. Fostel will talk more about the data, analyzing the data, and that's part of this presentation. Next slide, please. Thank you. You want to have a follow-up study just to make sure the study is reproducible. So you want to conduct a follow-up study using that same study design and procedure schedule. You want to maybe include one or more of the therapeutics or one of the more of the chemicals to know with a known efficacy. So you'll know that what to expect in the what to expect from that chemical, so you can use that information to see if that study is going the way it should. To ensure reproducibility, you want to use the same statistical strategy to analyze studies. the study that you did and then the study that you're repeating to make sure you're getting the same data. And if you have collaborators, ask the collaborators to do the study and then replicate your findings. And the ability to replicate the findings across laboratory settings will confirm the reproducibility of using TDB. Next slide, please. You want to, again, map out the data flow and get the help of a data scientist early in the study. You want to be sure that you're developing a place for data to be stored. And you got to think about where that data is going to be stored. It's going to be a lot of data, and you have to think about what your policies are for your institution. Can you store the data in the cloud? Do you have the bandwidth to store the data? For us, we were not allowed to use the cloud to store our data because it's our data cloud that belonged to another entity. So that's to the vendor. So you got to think of those things as you're doing this. And also, you want to engage early with your biostatistician because that will improve the study design. And data scientists and biostatisticians are not the same. And I believe Jennifer may be talking about that a little bit as well. So, next slide. Time management. You want to set realistic timelines and share with everyone on the team and the stakeholders. Be ready to be flexible, because as you're going forward in your study design, as you're going forward, you will see that little things will change, or you'll think of something that, oh, I didn't realize this. And I didn't realize this, so we have to build in time for this. And I will say that this will happen on your initial study, maybe your initial couple of studies, but as you become more familiar with using TDBs, you'll be more familiar with how to set up the study design. And I believe that's my last slide. Thank you, guys.
Thanks, Angela. I hope people can hear me.
Yes.
So Angela and I are colleagues, and we've been on this journey together, and it's been definitely a team building experience, and I've learned a lot. So my role at NIEHS is to support our data system, the chemical effects and biological systems that houses all of the data. I'm going to see if I can take this off there we go. So what is
Jennifer?
a digital bio? Yes.
I'm sorry, you're not sharing your slides, you're sharing the Teams
Oh, excuse me.
image.
Thank you. I should have asked. Where are we? Stop presenting. And then go to share. I thought I had the whole screen. Can you see this now?
Yep, that looks good.
Okay, great. So just a quick introduction. I apologize. Thank you. So I manage SEBS, and that's our legacy database. So what is a digital biomarker? Differentiating it from a biomarker. These have been around much longer and have been validated and useful for just an indication of some biological state or condition. But a translational digital biomarker are objective, quantifiable markers that are collected by means of digital. So they're clinically relevant, the translational part, and they're collected by means of digital technologies. And examples would be a video. They're non-invasive, a pulse Doppler, a wheel use, continuous collection in their home or cage environment. So in general, now there's all those methods that I just mentioned, there'll be data collection. And as Angela said, there's going to be file management. And then possibly moving the data files depending on their size. So all of these require a logistic consideration. And then we're going to reduce the analog content of the slide, the data, into a set of numbers. So for example, how far an animal moved, or was the heart physiological within normal limits? And what's exciting about this field is new. So unlike measuring a traditional blood clinical marker where you know the range, these may not have a fully standardized approach. They may not have a background range. And so it's quite an interesting time. So just for clarity, and I apologize, this does sound a little nerdy. The NIH has defined these different data levels. So data level 0 is directly off the instrument. Many instruments provide human readable files, so data level 1 and 0 might be the same. But in the waveforms, those are not human readable. They're a bunch of numbers. So we have data level 0 here. As Angela pointed out, they'll be coming off the instrument. Then they're converted into a human readable file. So this is data reduction. Then we clean up the data, take out outliers. They're normalized to data level 2. And then you get your biostatisticians to do some statistical analysis or other bioanalytical analysis. And so that's level 3. And then finally, they're integrated into biological context. So what happened in other systems that were comparable? What was the control levels? And then you get your conclusions. So these are 0 is the instruments, and I'm going to be focused at this level in this talk. So going from data level 0, remember that's the instrument data files. So the data files can look something like this. And what you are actually probably interested in is that the heart rate is 281 beats per minute. And so there's a lot of work to go from this into that, and you probably will need specialized software. And maybe there is no specialized software. You may need specialized methods. And it's really helpful if you get both a subject matter expert who can look at this and tell you from the point of view of the physiology what makes sense, but also from the point of view of the data science, how it gets from there to there. So there's different methods for doing this. So data mining is sort of exploratory data analysis, and then data science is to develop an algorithm. So for example, data science was used to look at astronomical dots of stars and identify that a comet NIEHS, was moving through this, and you could see that at looking at a time series. And similarly, data science can say, oh, that's obviously a mouse. And I can put them together and show that this is actually a mouse moving. And so that's the skill that's needed here. So you cannot do this without these experts. There's also biostatistics, which we all know and love. That's the, is there a trend? Is there a statistical significance? But we have machine learning also. So new and better methods to leverage data to improve. So for example, this machine learning takes into account, so it's looking at fruit flies walking around, and it can say at this point, these two are close enough to be considered touching. And it actually developed from the data how close was close to count as touching. So these are new parts of the data science world. Importantly, because this is what I do for a living, you're going to need to manage the data. So how big is it? Where does it live? What configuration? Blah, 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 blah. You probably have people at your institution who know the answers to these questions if you ask them. But these are going to be quite large and quite unusual data sets. So you really need to bring these guys in early. Particularly important is what is your plan for backup and data recovery? Because oftentimes that's you need a specialized plan for that. So again, you need to know where you're going to fit in here. So tier 1 is what most regular people like me work on. It sits in your computer. You can also get an external drive or a special capacity disk, or you can go to the cloud. And like Angela was saying, if you're working on the government, there's special methods to get you to the cloud. And you just need to plan ahead for that. So going back to this data level 0, again, the instrument files. So what is going to be your process for reducing the data? There's probably more than one way to do it. Maybe there's a standard way, maybe there's the vendor way, maybe there's public debate in the literature, and people have new methods that they can share. And as Angela said, how do you know which is the correct way to do it? There's quite likely to be interpersonal differences. If you remember that waveform file, depending on how you set your parameters, you might get slightly different. It may not be 281 beats per minute, but 280, which is correct. Secondly, you're going to have a bunch of files. Is there a public repository, and does your institute permit you to use that? Because you can deposit there under embargo, and then when you're ready to make it public, make it public. You might need to convert to an open standard, and oftentimes, though, the repository has analysis methods, so you put them there under embargo. You can use their standardized analysis methods. And again, data management is going to require expertise, and so you need to estimate the total size and make sure that you're working with your team to make sure, as Angela said, there's space to put all these files. So we're talking about study design here. So there's your normal study design, right? What are you testing? You're testing a drug. We're doing 2-ethyltoluene. There might be intrinsic variables like the sex or the weight of the animal, and define these, and preferably with a controlled terminology. But when you're measuring things, there may be specific confounding variables. For example, we recently, in another study, were measuring the acoustic startle response and discovered that one chamber had a small noise leak, and so the animal didn't have as robust a measurement. Is the time of day important? Important, you're going to be sending your data to various places. Keep the same animal identifiers. And then talk to someone who's done this data processing before. Do instruments have different variabilities? Maybe your 2 technicians might have slightly different parameters. And also consider how long it will take to do all of this work, because if you have work that requires an hour per file per day, and you have more than 20 files, that's going to be tricky. So before you start, sit down and talk to a lot of people. Like Angela said, what metadata do you need? So first of all, what is your scientific question? I just said that. You're going to know the answer to that. But talk to people. What possible confounding variables? Can you control them, or do you need to measure them? So ambient conditions handling is, like Angela said, is the technician in the room or not? And then what metadata about the technology will you need in order to do a reasonable data reduction? And then what is your data plan? Okay, so what type of data level will be generated? Do you need a tool or expertise to reduce it from the waveform to that 281 number? Is there a community standards, or will you be needing to develop that? Or what is your plan here? Where will you reduce it? So a lot of times, if you collect the data at the instrument, you don't actually need to move those massive files. You can reduce it to level 1 at the site of the instrument, which will save you a lot of time and trouble moving files around. So what size, how many files, how are you going to take care of them? Is there a public repository? And how long will you retain it? And just a little note, in the cloud, you can get the special Glacier storage, which takes a few days to get back from, but is not expensive. So are you going to keep your data or not? Big question. So a good strategy is to store the files where they were collected and then reduce the data there. Now, as Angela said, though, if you're working with a CRO and you reduce them at the CRO, you may not have real-time or even within a few days access to the data to look at the original data level zeros. Also, if you reduce them and store them there, you may have a storage cost. And then, as I said, a lot of times people share the code for their reduction and analysis, and that would be something you'd want to install with the data files. If you store them for a long time, obviously it's going to be expensive, and sometimes rerunning the experiment might be more cost effective. It may be petabytes of data. There's also the environmental cost of data storage. The cloud generates a lot of electricity. It uses, actually, and animal welfare. You may discover that when you reanalyze data that you kept from 10 years ago, something that you didn't observe at the time, didn't know at the time, may surface and cause a concern. Definitely, you want to retain it during the course of the study so that everything is handled consistently. And if you discover a systemic bias, you can go back and address it. If you retain it longer, your data are available for new methods, so people can work out new analytical methods, which is a plus. You don't have to repeat the work, which is very cost-effective, and it takes a lot of animals. And you can collaborate. You can take your data, someone else's data, and collaborate for a meta-analysis. Like Angela was saying, can it be reproduced? Can you learn more from the second time through? And you can collaborate with data scientists to improve the algorithms. So just in summary, this is our translational digital biomarker, which I thought was a very nice definition. And just to clarify, if you get a new data type, it's not going to be plug and play. You're going to need specialized tools, a lot of planning, and a lot of expertise. And new expertise you may not know. Don't just tell me it's big. You need to talk to your data manager and kind of try and quantify the size of a file. And then you need to identify what you need to reduce data level 0 to numbers so that you can analyze them. And you need a technology subject matter effort. Also look for potential confounding variables. Talk to someone who's done it for things I wish I had thought of when I did it, and try and utilize public resources if it's feasible for data storage and data reduction and analysis. And now I'm handing over to you, and I will stop presenting. Thank you.
Thank you, Jennifer. While I'm pulling this up, I wanted to introduce myself. My name is Ellie Carlson. I'm a veterinarian by training. Oh, we need permission to share your screen. Uh-oh. And I'm a veterinarian by training, but I'm working at a company whose imperative it is to leverage technology to understand the biology of aging and allow us to live longer and healthier lives. So bear with me as I go through my setup here. And I have been part of the North American 3Rs Collaborative as both a technology developer as well as an end user. And it's been immensely helpful for us to be able to share our experiences and sort of go through lessons learned so that we could accelerate the adoption of digital biomarkers or translational digital biomarkers. I'm struggling a little bit, Matt. I'm wondering if one of you could just share my slides or and progress for me.
Yes, Ellie, I am sharing right now.
Awesome.
So we are good to go. So.
Okay, great. So as a company that studies aging, we wanted to figure out if there was something that we could develop in-house to better understand the biology of aging. And so there are numerous brainstorming sessions, and we're talking about 20 people involving biostatisticians, data scientists, machine learning, computer vision, our aging researchers, our vivarium operations. And we wanted at a fundamental level to understand what is it that we're trying to measure. And in this case, it was distilled into a multi-phenotypic assessment of physiological decline with age. We wanted to be able to do frequent longitudinal measurements over long time spans. So for those of you who work with aging models, sometimes it takes years to see these phenotypes in decline in health. But we also needed them to be non-invasive, right? Like we couldn't have readouts that required serial large blood sampling. Oh, can you go back? And we also wanted to be able to simultaneously measure numerous parameters. So again, we had scientists involved, we had our data scientists, we also had our vivarium operations, and that includes our animal technicians, because we were really set on engaging and also expanding their role within the vivarium and within the research mission. And then we're also very lucky to have software engineers, our IT, and then data managers. This was incredibly important because our plan was to generate on the order of like decades and centuries worth of video data to develop and train these models. Next, please. So what biomarker will we try and replace? Many of you may not be familiar with the Frailty Index. This is one way that aging researchers in the clinical world measure decline of humans. And so there are a number of these Frailty indices on mice published, but the one described here is what is used the most commonly here at our institution. And as you can see, there's a number of systems that are described with various observations that you could note during an exam. And it's really a concept that helps explain the difference between chronological age and biological age, and it could be used to help us predict lifespan, essentially. And so the picture here is our setup in our behavior room that our technician uses to assess all of these various 31 aspects of health for each individual mouse. Now, as you can imagine, that's very, very time consuming and not that fun for an aged mouse. Next, please. So the frailty index is useful in that it produces a metric that shows a general health decline, and it lacks specificity, so there isn't a focus on any one particular system. But again, there's lots of big practical problems, right? It requires a lot of hands-on work by the observer. These studies oftentimes take years, and for those of you who run operations, staffing is a major challenge, and the turnover of research folks. You want to make sure that you have consistent staffing, and that's because the inter-observer variability is generally greater than the biologically relevant variability in the case of frailty indices. Next, please. So before we embarked in this journey, we really had to do a deep dive. Was there evidence that machine learning could be applied to aging and frailty scoring? And so the Sinclair group at Harvard put out a really nice study where they used machine learning analysis of manual frailty indices. So on the top figure, you could see that they measured frailty for mice starting at age 21 months to 36 months. And then they also developed in the figure below a survival curve to show the decline in survival. And then they used machine learning to take the frailty index and either predict the age or predict how long that animal would live. And so we thought this was a really interesting proof of concept that frailty index could be useful, and we could apply machine learning to that that biomarker. Next, please. And then we were asking, was there existing evidence that frailty scoring could be automated? And this semi-recent paper is from the Kumar lab at Jackson Labs. And they use computer vision to generate a frailty index from video of mice recorded in open field. And so this is definitely less invasive than the traditional frailty scoring. However, it still wasn't in the home cage. It still required a lot of technician time and researcher time. And so we wanted to take it a step further. Next. So we started to brainstorm a study design to develop a home cage digital frailty index. And these would be new digital biomarkers that we were developing in-house with our machine learning team. And some of the considerations that we asked ourselves when we were thinking of what features or digital biomarkers to develop was, can the new features be realistically observed using video? And would that type of frailty be likely to worsen with age? So we came up with this goal of developing a non-invasive automated frailty index based on computer vision on home cage video that will correlate with chronological age. And we had to consider what resources were available to us. So Calico had an aging colony of diversity outcross mice at JAX, also known as JDO. We had a home cage video rack system and software pipeline to store that video. But we also had some limitations. We only had 48 video cages, and our study would, to be appropriately powered, had over 200 animals. We only had one researcher, and we're intentional in using one researcher to perform those frailty indices. And then lastly, we only had about 9 to 10 months to both collect and analyze that data. And that's because this was a pilot study and proof of concept that was part of our annual goal cycle. And we really wanted to push to show that we could develop this digital biomarker and use it for future studies here at Calico. Next. So after hours of brainstorming with the full team, we came up with our study design. This is a photo of what our video cage looked like. We had both male and female diversity outcross mice. Again, we had over 200 of these animals across 8 age-matched cohorts. Our plan was to do a semi-longitudinal and cross-sectional study design, again, based on our time constraints. So the timeline below shows the age of the mice and the number of male and female mice assigned to each cohort. Every 6 weeks, we would take a manual frailty measurement, followed by a 1-week session recording in video. Next, please. And so during study planning, again, reiterating that we really had to have everyone in the room, there was a lot of questions before we landed on that study design. So one, should we do manual frailty relative to video collection? And so our aging researchers really felt strongly that we should do the manual frailty before putting them in their instrumented home cage. Which age groups should we collect video from and assess frailty? Because we had animals that were very geriatric, it was important for us to go in sort of reverse chronological order, where we were taking the oldest animals and looking at them first. We knew that there would be attrition throughout the duration of the study. And then working with our data managers, we had to answer the question of what kind of metadata was needed for analysis. And so in addition to age and sex, because we were working with a diverse outcross mouse, we wanted to know what the coat color was, because that could impact our measurements, as well as body weight, and then the frailty score. Other considerations that we had to talk about, things like how long do we need to acclimate them prior to manual frailty? How long did we need to record them to get a good signal on some of our metrics? And then lastly, this is where the data managers and the vivarium operations and animal technicians really worked hand in hand, is how do we track the location of all the animals with their identifiers in their cages? We were actually moving animals between single housing to group housing for our female cohorts. And so before we did the real study, we actually did dress rehearsals within the vivarium to make sure that animals were being moved and tracked appropriately. Because if you put the wrong animal in the wrong cage, you're essentially collecting data from the wrong animal. And so we found it very critical for us to be able to do this without making any mistakes. Next. Great. So these are the results that we published. You know, we did the manual frailty on all 200 plus mice, and this is the curve that we got, and we had a correlation of manual frailty index and chronological age of 0.27. Now, it's not the greatest R coefficient, but what's interesting is that for JAX-DO mice, because they have variable phenotypes, we don't get as strong correlation for some of these manual frailty components. Things like coat condition or alopecia, for example, are more represented in Black 6 mice than DO. Next. Now, when we developed our digital frailty index, the digital biomarkers that our machine learning team developed in this short time period reflected various domains. So body condition, environmental engagement, physical capabilities, so mainly gait speed, and circadian rhythm. And so that represents certain activities and how closely tied that is to the light-dark cycle. And what we found was that our digital frailty index, that's the figure to the right, had a higher correlation to chronological age than, that's the figure to the left. Our digital frailty index had a slightly better correlation with chronological age. But when we combined that with our manual frailty index, when we took both into account, we had an even higher correlation to chronological age. So, you know, for a first pass of all of the new metrics that we developed, it was not as strongly correlated as we would have wished, but it was still better than the noisy manual frailty index. Go ahead, Matt. So these are some of the figures from our paper that show, how we came up with our various digital frailty parameters and how they performed individually to chronological age. And there were several lessons learned from this version 1. Again, some of these machine learning models we thought would correlate really well with aging based on traditional frailty data were not as informative. So I mentioned the black 6 coat condition was one of those very strongly correlated measurements in frailty, but in the DO mice, it was not. And then we also built a nest movement parameter, but we did not observe a very strong signal as animals aged, and only really saw that parameter change severely when they had health concerns. So when you go upon this journey, you'll have hypotheses of what will be informative for your specific model, but be prepared for those for your study and your pilot work to tell you differently. The other thing that was very important to us was doing computer vision on a diverse outcross or diverse phenotypic outbred cross, was so that we could have our models trained on a wide variety of phenotypes, so that way we weren't just training our models on black 6 mice. So we could have various inbred strains and outbred crosses used in this system for future studies. And next. So all in all, the scientific insights from our first pilot study was that our digital frailty index correlated with chronological age, and it performed slightly better than our manual frailty index. And that when we combined these two, manual and digital, we had an increased correlation. So next steps, we might consider adding other non-digital biomarkers or other assessments to the frailty assessment. And there was definite 3R's impact. In the future, we'll have reduced handling of our mice and have them housed in an enriched environment and reduce stress of doing behavioral assays. And then operationally, we could run these studies without research staff in less time. However, there is still a lot of work to be done. This was just our first pass. We recognize that we need to refine the existing digital biomarkers. Now, I think a lot of folks will be like, well, maybe you could look at another biomarker, but it's also really important to note that developing new biomarkers is very, very resource dependent. And that's really leaning heavily on the computer science and computer vision folks. And so that resource is really, really hard to come by. And so we're going to have to consider what other non-invasive biomarkers we're using in-house, whether that be imaging, or if we combine it with biological biomarkers, things like glucose tolerance, to improve the power and predictive capacity of this frailty index. Next. That's it. Thank you.
So thank you so much to all the panelists and giving a presentation. So now if you have questions, please put them in chat. We would definitely appreciate those. But maybe we can kick off with some of the questions that came up from the audience when they were registering. So one of the questions is, excuse me, are there effective systems or procedures to apply existing biomarkers for application with novel oral delivery vehicles? And Matt, I guess if we could put that panelist on the screen, that would be great.
Could you repeat the question? I'm very sorry.
Sure. So the question is, Are there effective systems or procedures to apply existing biomarkers for application with novel oral delivery vehicles? I mean, I guess I can take a stab at it. I'm not really clear on the question, so I don't know if Karlsson is online, if you could expand on that in the chat. But I guess it depends. We always talk about the context of use, right? So depending on what you're trying to measure with all the delivery vehicles and which vehicles you're utilizing there. That's the only comment that I guess I can add. All right, so next question is how can translational digital biomarkers be leveraged to inform earlier humane endpoint decisions on studies where there is a high potential for pain and distress, such as sepsis or vaccine studies where the animal is challenged with organism?
But I mean, I think there's definitely a lot of potential. I mean, can't remember exactly the paper. It wasn't a translational one. It was a sepsis model with the temperature measurement, and they noted that prior to there was a temperature drop off right before a full blown kind of a move into a morbid state. And I think that is a wonderful aspect of these biomarkers, the potential to do that, to look for things that we typically could not see in our normal clinical ops. And that's actually what we're kind of hoping to do. We already know what our toxicant is. We're hoping to look a little bit ahead. Like, is there some change in respiratory rate? Is there something that's going on prior to that we can pick up and kind of use in refining our humane endpoints?
I would say that that would have to be model specific. You know, there would be a pilot study where you're comparing the digital biomarker, whether that's movement or respiration, to your current clinical scoring method. And so, because every institution's humane endpoints are different, and depending on what outcome you need for studies when animals are being challenged, yes, I guess the short answer is yes, you can use the digital biomarker. But again, this pilot study, in collaboration with your vet, your researchers, will all have to be done to determine whether that digital biomarker is an appropriate replacement for your traditional clinical endpoint.
Or not even a replacement, but an enhancement to your traditional to what you do traditionally in your study. So I think that's what we're really want people to see, that digital biomarkers can help enhance the studies that are being done from an animal welfare point of view.
I mean, I can also add, so when we did a liver, there was a publication, a lung fibrosis model, and with the digital biomarkers, we're able to see the changes in activity with the animals sooner than we were able to see with the traditional digital biomarkers. And also those changes aligned with histopathology endpoints as well. Obviously there, additional studies would have to be done. Can we to confirm that, and then can you replace, for example, the histopathology endpoints? Not all of them, obviously, but some of them with this digital biomarkers that were assessed there. So the next question is: are clustering algorithms in use to identify groups after data collection, and how are these integrated into study design?
So that would be a data exploration that would be carried out whenever you have a new and large data set. So you want to look for any underlying structures. So clustering would be helpful. And And that will also help you identify significant outliers. So yes, that would be part, but after you've gotten to the numbers, clustering is better in my experience with numbers.
Thanks, Jennifer. So then the next question is, there's a potential problem with tracking behavior of individual animals when they are housed socially. Will the digital solution require single housing or tracking identity and comparing behavior of different individuals in the same cage?
I don't have much to add, just to say it is a challenge for us. We end up having to do individual tracking simply because of that. And I like to hope as things develop and more tech providers move forward that we'll get to that point. But right now, that's one thing we're definitely struggling with.
Yeah, I would say that for our studies for computer vision, the state of our technology is that we also have to single house. There are digital biomarkers available non-video that do allow group housing. And so this is again where you have to get back to your researchers and your model to say, well, what's important to us? Do we need the animals to be group housed? And what do we want to measure? Because if you just want to measure gross activity, there is technology that exists that allows for group housing. And so, there's, just to reiterate from past talks, there's no one size fits all solution. And so you really have to prioritize your housing conditions, really understand what's going to impact your model, and then make your technology decisions based on that first. It's very difficult to buy into a technology solution and then try to make it fit your research after the fact.
So the next question is, with so many repeated measures with translational digital biomarkers, how do you manage the false positive rate? And how do you redesign to accommodate for a high number of data points?
So that is where you need a biostatistician to tell you that. So after you've reduced it to numbers, if you're measuring every day, you do need somebody who can do repeated measure analysis and get that question answered.
So the next question is, please remark on quality of data and translational components in comparison to studies that have not been done using translational digital biomarkers. I'm not sure. So John, I think you're here, right? If you want to expand, because that's a question that came from you. I don't know, maybe if you can clarify that for us in the chat. Or Matt, do we have the ability to unmute John?
Yes, I should be able to do that. Let me find him. All right, John, you should be able to unmute.
So while we're waiting for John, the other question is, or even I guess a comment, also on biomarker validation and validation test design. So I know maybe you guys can comment, how are you validating, your biomarkers? And you guys talked about that a little bit, but maybe if you can expand on that.
Yeah, I think the information is available on our publication on Frailty at least, but we had to run a side-by-side study, and it was massive and very intensive to compare our digital biomarker to our traditional biomarker. Could we have collected more sort of biologically relevant information, blood samples and whatnot, to help validate? Yes, we could have, but given our staffing, the timeline, and narrowing in on what we wanted to validate, we did not include those things. So again, it's almost always a side-by-side comparison of your traditional biomarker to your digital biomarker.
I agree. We haven't gotten that far yet. We've done our traditional, and now we're moving on to the use of the biomarker. But yeah, agreed. It's just trying to line them up and kind of replicate the study and as closely.
No, and I guess if I can pitch also, so on the publications from the group that I shared in the chat, we do address briefly the verification, validation, and qualification of digital biomarkers. But our next manuscript that we're working on is specifically on that topic. So we're going to be expanding on that, and hopefully before the end of the year or early next year, so you will be able to see that. But also, if you are interested in participating in an effort, please join that group and let us know, and we can add you to that initiative, to that subgroup. You just see there's more questions. So this is a question from Theory. And I know, Theory, you're here. I'm going to unmute you as well. But the question is, patient data and clinical research, such as biomarker qualification, are key steps in drug and vaccine development. What are the outputs and values of, in quotes, human models? I'm not sure if I really understand that question. But you're just unmuted, Theory, if you want to jump in and maybe expand on that. And I guess so maybe I can take a stab at that if I understand that correctly. So there's digital wearables and other technologies, health monitoring and patient monitoring technologies on the clinical side that we use, starting to use pretty commonly. And there is a validation or a biomarker qualification process in place through regulators like FDA, for example. And that has been, I guess even there's guidance on it that came out, I believe, last year or 2 years ago, how the process works. For us on the preclinical side, that's what we're trying to adopt as well, but it is slightly different. And there's also a nonprofit organization on the clinical side that has developed a regulatory roadmap for the technology providers, how to actually get approval for various biomarkers. But again, those are the organization is called DIME, and I'll put that in the chat here as well. But again, that's very context specific. And John, you're not able to turn on. Let me just try it on my end. Just do it one more time. All right, so you should be able to unmute yourself, John. And so as we're waiting, I guess maybe a question even for me, for the panelists, if you guys could go around the virtual room and say, so what are some of the most frustrating things that you have faced? And I'm sure maybe you can have a list, but maybe identify one or two, and how were you able to address when working with these projects with the digital biomarkers? So Ellie, maybe we can start off with you.
So the most frustrating aspect of working with digital biomarkers?
Yes.
On the operational side, it's that data management and making sure that everybody understands the importance of, where does the mouse move and how is it identified? Because if you don't get that right, then you can really mess up your data. And it's very, very difficult to untangle once a mouse is placed in the wrong cage, for example. So there has to be a lot of back end fixing. So that's very frustrating. And I would say the second thing from a scientific perspective is having our scientists work on developing a biomarker and then to have it not work out, not be as informative as you wanted it to be, and have to go back to the drawing board, because again, it's very time intensive to run these analysis. But all in all, I would say that the benefits and the joys of seeing that transitioning to a digital biomarker is better, in terms of correlation and staffing, outweighs the frustrations.
And so Ellie, if I can just to follow up on that on that comment about the expectations, right, when you're looking for digital biomarker. And I mean, is it just because the expectations are way too high, right, for what we're trying to identify, or is it just that the technology is not able to potentially just do what we're asking it to do?
I don't think it's the expectations. I think it's that we just don't know what we don't know. And so when we look at an old mouse, we go, oh, obviously an old mouse has poor coat condition. You don't realize that on camera to video, the camera doesn't see that as well as we do. Or maybe we didn't develop a model that can assess it as well as the human eye can. So we just, again, I think it's just for lack of knowing. This is like uncharted territory. And so I think we have to prepare ourselves and have that growth mindset, right? Like prepare to be disappointed, prepare to be surprised, but take it as a V1 and then go back to the drawing board, bring all the people in the room again and try to make something better.
All right, thanks, Ellie. So Sheba, maybe we can turn to you. What's one or two very frustrating things? How you address them?
I think sometimes just explaining what you're trying to do to your stakeholders, because sometimes there's a lot of background in explaining how the tech works. And I think one thing, and Jennifer and Angela can attest to this, multiple meetings of just showing this is what we expect, but I do not know for sure this is going to happen repeatedly, and just trying to get their buy-in or confidence to understand that this is new and you're trying to show that this has potential to add to the knowledge that we have. But additionally, in some instances, going through, okay, well, this is how this works. This is starting from different levels of educating people based off what they have and know in their background. So that can be definitely a challenge.
So Jennifer, we'll pass over to you. What's the most challenging thing?
So we are still on the mountain looking over the uncharted valley of the experiment. So for me, I'm not finding it frustrating at all. I'm thinking it's like, oh, cool. This is a technological challenge. Let's try and think of everything. It's going to be frustrating in 6 months when we do it and we realize what I didn't think of. But right now, I'm optimistic. So sorry, not much help here.
No, thank you. And then Angela?
All of what they said. I think the biggest thing is you don't know what you don't know. And for the study designing the study and working with the team, I think for me, the most frustrating part of it is the timeline. It takes a lot of time, and you are working on designing the study and putting everything in place, and you innocently ask one question that spirals everything out a different way because you didn't think, how are you doing XYZ? You didn't think about that when you were spending all the time with your team working on the study. You finally asked that question to a collaborator or the vendor or someone else, and then you get an answer that you did not expect, and you have to go back and say, we need to rethink this just a little bit more. So for me, it's it's just the timeline of, I think we're going to start on this date, but we learn new information. And so we need to incorporate that and move our data a little bit more. So again, it's really, you don't know what you don't know until you start doing it. So I guess the bottom line is you gotta jump in and do the study and then kind of figure out what you learned and what you didn't know. So this is why the pilot study is so important. Our pilot or feasibility studies are so important to do with this type of technology to help honing your focus on what you really need to know, what you're looking for, what questions you're trying to answer.
Great, thanks, Angela. And Matt, if you can bring John over.
Yep, so I have made John a presenter. John, I think you can unmute now.
I think I can, and thank you. Can you hear me?
Yes, we can.
Okay. So the value that I was talking about was trying to identify where the highest values we've seen so far in the digital biomarkers, and then where the greatest returns have been to date. So it's kind of a global question, and I think we can build on that global question in terms of where things have been moving in relationship to that. It depends on what system you're looking at and also what disease process you're looking at in relationship, obviously. But I think there have been great inroads, and I just wanted to hear from this panel, which is an excellent panel, by the way, where they thought the greatest values have been.
Yeah, thanks, John. So this time maybe we'll start, Angela, with you.
Well, that's a little bit difficult to answer because we haven't actually started our study. We're still we haven't started we know we're going to do a pilot, but we haven't started our study yet. So it's a little bit hard to tell. I believe the information that we're going to get from the study is going to be very valuable, but I don't know until we do it.
So Ellie?
For us, operationally, again, to be able to run an aging study where we are not dependent on one person collecting the data and introducing that inter-observer variability. But also company-wide, we have an imperative to utilize advanced technologies. So we have various cores, we're looking at genomics and imaging, and how can these digital biomarkers interface with some of those other non-invasive biomarkers such as imaging to increase the power of the insights that we gain from each in vivo study. So it's really, it is 3 Rs in that we're refining, but we are also just maximizing the amount of data that we collect and information that we gain from every in vivo study, I think is the big sell for our institution.
Thanks, Ellie. Sheba?
Yeah, I mean, for us, we were working towards it, but one thing I've noticed, just the animal welfare portion of it, just the ability to kind of look at the study and see certain points with the data that we're getting in, the potential for this being used. One neat thing is because it's a digital, the data is, obviously digital, but our statisticians like, okay, well, I have these numbers, I can drop this down because I have everything here. I can recalculate with less animals. I have the potential of this data being poured into different efforts to, reduce, refine, replace is just tremendous. And I'm really excited about that. So.
Yeah, thank you. And I guess because just a few minutes left, I guess if you guys had to give one piece of advice for somebody that's just thinking about engaging with this technology, what would it be? And Jennifer, maybe I'll start off with you on that one.
Yeah, very good. I think everyone said it all the way across. Get a team together, ask questions, ask more questions, and really, it's a fun time, so enjoy it. But yeah.
Thanks. Angela?
So I'm actually going to take a page from Jennifer's book and say, have fun. You know, just really start thinking about what you're doing. And we may make it sound like it's frustrating, but have fun because, you know, you're scientists and you like doing studies. So yeah, that's what I'm going to go with.
All right, thank you. Ellie?
I would say that it's very important to have a champion and to build sort of strategic allies within the organization, because no one person can do this alone. No one person has the background or the knowledge to really make this happen. And so here at Calico, I'm a veterinarian by training. I run the operations. I can make the business case, but I need in vivo researchers to make the case that this is better for them. I need the folks on the computing side and machine learning to find this exciting and interesting. To motivate them with 3 Rs, to collaborate across the organization with other technology developers. So a lot of it is this team building. And having a champion and having strategic partners who are as enthusiastic as you are across the organization really helps move these initiatives, which are very resource intensive, both in monetary value as well as headcount. You need all of those partners to champion this across the finish line.
Thanks, Ellie. Sheba?
Yeah, so it's similar to what Ellie said. You're not alone. You have this group. I mean, a big part of why we joined was to learn more, to participate, because I really think this is exciting and I don't know what I'm doing. So it's also nice to have people around me who I can go to and kind of learn from their experiences. So don't feel like it's a big project, but don't feel like you have to take this on alone. There are other people who've kind of walked ahead of you that you can, tap into their resources. So.
No, thanks, Sheba, for mentioning that, right? And that's one of the biggest aspects, or one of the many aspects for the North American 3 Rs, right? Is that we can learn from each other and work together for the animal welfare and the 3 Rs. So with that, I also want to mention for the next webinar that we're going to have is actually going to talk about the cost of engagement with these technologies and the return on an investment for these technologies. So I think that's something that we all have to address, right? When we're proposing to engage with technologies, when we continue to engage with these technologies. So again, I hope that you will be able to join us for that webinar. Again, we really want to thank all of the sponsors for the North American 3 Rs. I mean, none of this could really happen without them and supporting the North American 3 Rs Collaborative. And again, if you're interested, if your company organization is already a member and you want to just come in and maybe listen to one of our sessions that we have on a monthly basis for this Translational Digital Biomarkers group, please let us know and reach out, even if you maybe want to just try it out. Or if you like to connect with some of us offline, also please reach out to North American 3 Rs as well, and you can do that on a website. So again, I definitely want to thank again Matt for organizing this and helping to manage this. I want to thank all of the sponsors. I want to thank for the participants that join us for this. I know everybody is busy, so taking the time to do that. And of course, I want to thank Sheba, Angela, Ellie, and Jennifer for giving their time here. And again, here's also thanks, Matt, for the email. So if you have any questions, any comments you'd like to join, please do that. And I don't know, Matt, if there's anything else that you wanted to add. And Matt, you're muted.
No, I don't have anything to add. That really wraps it up nicely. Thank you again to all of our guests for joining us. This is such a cool topic to be discussing and really discovering new things about. Thank you to the panelists, and thank you, Shapan, for hosting.
All right. Thank you so much, everybody. I hope you have an awesome rest of the week, and we'll see you at the next webinar.